Well, this is my YRI presentation on stem cell fate using machine learning models. And this is presented by Shaswat with my mentor, Dr. Senthilkumar. So the idea behind this article and the presentation was to create a machine learning framework that could design and predict different stem cell differentiation types using pre-existing data structures. And the purpose of it was so that we could accelerate lineage-specific therapy and regenerative therapy model. It reduces lots of trial and error when considering different types of differentiation among stem cells. The machine learning model was created around a random forest with different steps that can identify the different kinds of stem cells that were in the data sets. And using different biomarkers, lots of genes were created and they help drive the model predictions, and you can validate them using the experiments from the machine learning model. So the purpose of the machine learning was to integrate it with other single cell omics in order to enhance stem cell differentiation. Traditional differentiation between stem cells kind of tells us what changes are impacting the stem cells, but it doesn't really show how those changes can make tiny different decisions in stem cell fate. And machine learning specifically is really good at identifying those non-linear models in the dimensional spaces, which can help you predict the future outcomes of different types of stem cells. So by kind of integrating like the different single cell insights, like the marker genes, into this big pipeline that can filter out through different types of stem cells, you get the best of both worlds. You get a very cost-effective way to collect data, and you also get a very high resolution output of what the data is showing you through stem cell differentiation. So just going back to methodology, there was a bulk of RNA sequencing I collected online in order to put it into these data sets. So there was first, going into the model, there was lots of pre-processing, filtering out data and normalizing it. And ultimately, there was 4 main different kinds of sets that the data was split into, and then it was all CD90 and CD49, and then with different polarities like plus plus, plus minus, minus plus, and minus minus. So it kind of split the data into 4 different sets to show a different output of the differentiation. And going back to pre-processing the data, it was really important to filter out and correctly identify quality control, so remove all the low quality stem cells, and also apply a different factor sizing to correct the library differences, as well as stabilizing the data sets for different methods. So going back to more analytics, this just talks about the different kind of gene features, like biomarkers, other important outputs that the stem cells were able to find, and also just overall kind of identify the different performance levels of the different stem cells to figure out how useful they could be in real life regenerative medicine. So using the machine learning model, there was 8 main plots that came out of the results summary, and the analysis using these different kinds of techniques like PCA, TSNE, and UMAP, they all kind of confirmed the different lineage clusters that they had, which kind of is the entryway into showing how effective machine learning could be in predicting different types of stem cells and their differentiation abilities. So first is PCA, which is principal component analysis. And what it does is it splits the 4 different kind of subgroups into different clusters. And then it's split along 2 axes, PC1 and PC2. So PC1 kind of reflects how it occurs with differentiation, how effective it is. And then PC2 is how effectively the biomarkers get captured on the surface. It highlights how the principal component analysis can reduce all these thousands of genes into two main like axes through these four subgroups. So you can kind of identify the different kind of differentiation that they have. The next kind of subgroup that came out of the machine learning pipeline was T-distributed stochastic neighbor embedding, or and what this does is it creates like these little islands for the blue and yellow subgroups, which indicates how similar they are in transcription ability. And the kind of overlap between them that they have reveals that it's more of a program that the stem cells are going through, and how you can display each sample onto like this graph, so you can identify which kind of cells have similar gene expression between them in order to identify the different types of subgroups. After that, in the results section, from filtering out these stem cells, you also got UMAP and a correlation heat map. So UMAP as a whole is pretty similar to TSNE, but what it does is it balances like the different structures that they have, and in a kind of trajectory, like almost like slope, rather than just like different like islands or subgroups. And this kind of gradient kind of interprets how the stem cells change with time, in order to best resolve like the different clusters that they were bringing, and how they can transition from a more like early primitive state to a more differentiated state after the stem cell. The fourth one was top gene correlation heat map, and it just took like a variety of different genes, like, and how they correlated with one another in a gene pair relationship along these axes, and it just showed and it just kind of went through like how they reacted amongst one another. And then I think after the second to last two, the random forest with biomarker genes and the cross validation. The random forest feature plot is especially important when considering how effective these genes were in differentiation. So it shows how only like a few genes, like the top one here, ENS 37441 and ENS 273805, these two genes had really, really strong differentiation predictions from the machine learning model. This is really important when considering like what kind of future insight could I bring to this? How could these like specific biomarkers like more readily influence stem cell differentiation? Also, this is another kind of heat map with threefold cross validation, and it just shows identification amongst these types of groups, and how like mistakes happen when occurring with stem cell differentiation. So where does the model work really well? How effective is it? And where does it kind of need more data? So it kind of fills in like the gaps in future exploration. Finally, the last two were different kinds of curves, ROC curves and PRC curves. The ROC curves were especially important. The ROC curves were especially important for understanding like where the genes fall below these lines. If it falls underneath this curve, it means that the probability that a randomly chosen positive cell is higher than a negative cell, where if it was above, it would be the opposite. It's really predictable at identifying certain types of cells, like CDNO plus and minus, the yellow line. It's really effective at identifying that type. But the other ones, such as like the double minus, have a much lower AUC, which is just area under the curve, and it needs more data and more like performance to identify. On the other hand, precision recall curves identify like a similar aspect, but the thing is that they're more informative than ROC curves, because the positive class is pretty rare amongst it. So the orange curve has extremely high precision, at low recall, which means that when the machine learning model is confident that it's correct, it doesn't very often make a mistake. But a lot of the times it misses the true positives, and it requires more data to back up the evidence in how effective these curves are in improving precision and recall. So going back to the model and the strengths and limitations of it, random forest and the different types of identification tie directly back to genes, and they don't really have like any kind of filtration between them. But the problem is there is a huge data constraint within like such a project like this, where you couldn't research and collect your own data, and using these public data sets, there was some limitations behind what the machine learning model could filter and get out. But ultimately, integrating that kind of RNA sequencing data allows you to interpret it and create your own identification when pursuing like a research project like this. So the kind of overall conclusion is that machine learning bridges are really important for kind of bringing the computational side and also the biological function that RNA sequencing data sets have towards a project like this. And it's really important to combine different types of data and continue exploring in order to further identify data sets and kind of pursue and take on a future aspect towards this. So that's how you can incorporate more data and integrate more layers for better insights in stem cell differentiation.